Hi, my name is Anri van Wyk and I'll be chatting to you about genetic management guidelines to guide the translocation of southern African vertebrates. So, one of the cryptic drivers contributing to biodiversity losses is integressive hybridization and reverse speciation. So what is hybridization? It is the interbreeding of individuals from different genetic populations regardless of their taxonomic status. Whereas admixture can then be classified as the mixing of previously diverged or isolated lineages. Hybridization can then further be classified as either natural or anthropogenic in origin. Natural hybridization plays an important role in speciation and the maintenance of biodiversity. The Virgin River roundtail chub is an example of a hybrid taxon that appears to have originated from hybridizing in the Pleistocene long before any human influences in the Colorado River system. Anthropogenic or human-induced hybridization can contribute to species extinction and may occur if there is a change in the abundance and distribution of the species. For instance, the removal of some barriers will cause restricted species to expand, and also the introduction and translocation of species outside their former distribution ranges can lead to hybridization. Anthropogenic hybridization can also be intentional and be used as a tool by conservation agencies. Intentional hybridization or genetic rescue has been used to successfully recover the Florida panther, where their numbers declined and the occurrence of genetic defects increased, so they decided to introduce 8 female panthers from Texas into the population in the mid-1990s, and subsequently the Florida panther population increased in number and ranges. Genetic rescue may have large improvements on fitness and evolutionary potential of small inbred populations, and it can become a very powerful conservation application if there is a low risk of outbreeding depression. Allendorf et al. Categorized anthropogenic hybridization into 3 types. Hybridization without integration, which produces sterile offspring. This type of hybridization may not be a threat to either species. However, it may slow population growth due to wasted reproductive efforts, and it can eventually lead to extinction if a threatened species is involved in hybrid mating with a more common species. Then widespread integration and complete integration is where the hybrid offspring is fertile. With widespread integration, both pure and hybrid individuals exist. However, if conservation measures are not taken to stop hybridization, it can lead to complete integration, where there are no more pure individuals left, only hybrid individuals. The conservation challenges of hybridization is the fact that there is no single policy or guideline that can be applied to all hybridizing species. And conservation and management decisions are normally case-specific, which brings me to our genetic management guidelines and how it can be applied to blue and black wildebeest. Black wildebeest is endemic to South Africa, Eswatini and Lesotho, and they have been introduced into Namibia and Botswana. They've been brought back from the brink of extinction in the early 1900s, whereas the blue wildebeest occurs throughout southern and East Africa. In South Africa, the historical ranges of both species are largely distinct, with black wildebeest normally restricted to open grasslands, while blue wildebeest are normally associated with a wide variety of savanna habitat types. However, the geographical ranges of both species did formally overlap in a number of areas. There are no documented cases of natural hybridization, and it was assumed that the differences in habitat preferences and behavioral differences of the species would result in reproductive isolation. It was only in the 1960s where hybridization between the species was first reported, and only later it was observed that the hybrid offspring is fertile. And during that time, black wildebeest have however been widely translocated within and outside of their natural distribution ranges without taking hybridization with blue wildebeest into account. Therefore, admixed individuals may have been used as founders of new black wildebeest populations as well as mixed with pure black wildebeest individuals. So for the black wildebeest guidelines, 3 scientific articles have been used in compiling the genetic management guidelines, and the markers used included mitochondrial DNA as well as microsatellite markers. There was no ESUs or MUs that was revealed, and the following management guidelines is suggested for black wildebeest. That black wildebeest should be managed as a metapopulation. If both pure and blue and black wildebeest were kept on the same property, all animals should be genetically tested before translocation to ensure no hybrid individuals are moved. Also, there should be no imports or exports from and to Namibia and Botswana, and black and blue wildebeest should not be kept on the same property. For blue wildebeest, 5 scientific articles were used in compiling the genetic management guidelines. The data included allozymes, mitochondrial DNA, and microsatellite markers. There's also no ESUs or MUs for blue wildebeest, but the 5 subspecies that have previously been described still holds, and the following management guidelines were suggested for them. That blue wildebeest should be managed as 5 different subspecies with no imports of the other 4 subspecies into Southern Africa. If both blue and black wildebeest were kept on the same property, all animals should be genetically tested before translocation to ensure no hybrid individuals are moved. And then also blue and black wildebeest should not be kept on the same property. We also compiled genetic management guidelines for the following species where hybridization might be a threat. For the hartebeest, 2 ESUs were identified, the northern and the southern ESU. And within the southern ESU, 2 MUs have been identified, the red hartebeest and the Liechtenstein's hartebeest. There is a threat of hybridization between the 2 MUs as well as between the ESUs. Therefore, the guidelines suggest that captive bred individuals and those on private ranches should be genetically tested to prevent inadvertent mixing of the ESUs or MUs. There's also a concern of hybridization between red hartebeest and blesbok as well as red hartebeest and tsetse. Hybridization between these different species occurred due to a lack of conspecific mates on properties where both species were kept. For impala, 2 ESUs were classified, the common impala and the black-faced impala. There is a threat of hybridization between the 2 ESUs. The ESUs have been geographically isolated. However, translocation events have brought them into secondary contact, creating a hybridization risk. The guidelines recommended that black-faced impala be genetically tested to confirm purity and that there should be no new introductions of the one ESU into the range of the other. And also there should not be mixing of the 2 ESUs on the same property. For sable antelope, 5 ESUs have been identified, which corresponds to the previously defined 4 subspecies, and then a new suggested subspecies. The guidelines only focused on the southern ESU, which consisted of 3 different MUs. There is a threat of hybridization between the ESUs as well as between the MUs. Therefore, the following management guidelines were suggested. There should be no imports of sable from countries containing the other ESUs. There should be no translocations between MUs within the southern ESU, and captive bred animals and those on private ranches must be genetically tested before translocation to ensure no admixture between the ESUs. And then another hybridization threat to sable is hybridizing with roan antelope. So the different outcomes of hybridization for red hartebeest and tsetse, red hartebeest and blesbok, roan antelope and sable antelope all produces sterile offspring, which will not lead to complete integration. However, as explained in the categories of hybridization, it can slow population growth due to wasted reproductive efforts, and it can be problematic if a threatened species is caught up in hybrid mating with a more common species. And then for the black wildebeest, sable antelope ESUs, common impala and black-faced impala, and the red hartebeest and Liechtenstein's hartebeest are all suspected to produce fertile hybrid offspring, which can lead to widespread integration, and if nothing is done to complete integration, where there is no more pure parental species left, only hybrid individuals. So in closing, the genetic management guidelines will ultimately contribute to preserving the genetic integrity as well as in conserving high levels of genetic diversity in local populations. Working hard for something we don't care about is called stress. Working hard for something we love is called a passion. Thank you very much.